to hear from all kinds of disciplines, which is great, great for us. Remember, you can see all of the recordings, previously. They're all on our website. I'll put that in a link to that as well. And if you have any new, if you have people that you'd like to see give a talk here at any career stages, are welcome. Disciplines are welcome across the board, from geophysics and seismics to microbiology, as we have today. So you can use the form that's going to be in the chat, and so feel free to nominate people for our speaker series. With that, I'd like to welcome Dr. Jessica Labonté to this month's speaker series. Dr. Labonté received her bachelor's and her master's from Laval University and her PhD from the University of British Columbia. She was a postdoctoral research at Bigelow Laboratory before she headed to Texas A&M Galveston, where she's currently an associate professor. Her research focuses on determining the role of viruses and their hosts in the various environments, including the subsea floor, as well as vents and ridges, and of course the aquatic environments as well. So thank you, Dr. Labonté, for joining us. And if you have any questions, feel free to use the chat, or also you can, at the end, unmute yourself or use the raise the hand button. We welcome all kinds of participation.
Thank you, Brandy. And yes, as Brandy said, I'm typically a viral ecologist, but today I'm going to talk to you about a small study that I did using the Novel Project Idea funds from USEP with IUDP, which was to identify and manage ubiquitous subsurface contamination. I'm aware that not everybody's a microbiologist here, but microbes are literally everywhere. So if you have sailed or sampled with a microbiologist, you probably that we're crazy because we wear a mask, we wear gloves, and we're like, don't touch this, like spray alcohol on everything. And that's really because microbes are everywhere. And this is just an example of microbiology 101. The first lab that we do is highlight how microbes are ubiquitous in the environment. And we have students wash their hands or not wash their hands and look at the microbes that are on their hands before and after washing, on their skin, their saliva, and then they're allowed to go all across the building. And we have these petri dishes that the agar is coming out of it, and it's like a little stamp, and they can stamp any surface they want, and they can see that there's always microbes that grow on everything. And so microbes are ubiquitous. I already told you they're everywhere on our body, but they're also everywhere we look for. So if we're thinking they're in soil, there can be 10 to the 15 cells per meter square, like on the vegetation itself. In the ocean, 10 to 12 cells per meter square, even in ice, and also in the air. It may not be as many. But if we compare how many cells there are in the air to how many cells there can be in subsurface samples, it's pretty close. So here we have a study that was done in 2012 where they counted like how many prokaryotic cells were in either in the Pacific Ocean at using different IUDP samples. And they found that, well, it's actually not that much. It can be 10 to the 2 per centimeter square to 10 to the 9. And it varies by where the sediments were located, as well as how deep they were. And usually the deeper we get, the deeper, the fewer cells there are. And why does this matter? If you have very few cells, 10 to the 2 per centimeter square, that's 100 cells. And there's pretty much as many cells in our sample that there are in the air around. So we have to think about how are we going to process our samples. And this is sediment. And if we think about basement samples, it's even less, 10 to the 2, 10 to the 4. And these are pretty much usually, sorry, I'm losing my pointer. They're pretty much close to the limit of detections of cells. So knowing that there are cells pretty much everywhere, how can we limit contamination when there are as few cells in our samples as they are in the ambient environment on our cells on pretty much everything? But before we start like figuring out how we can limit it, let's look at the life of a sample that so I'm more familiar with scientific ocean drilling, so I'm gonna base it on this, but pretty much any drilling or any subsurface, we have to deal with the same problems. But if we have a sample, the core will come on deck and we get our little chunk, our little whole round, and like if we're going to use it for microbial analysis, usually we take the whole round and put it in the freezer. And here I put numbers on how many steps does it take before the sample is properly preserved. If we take the whole round, we freeze it, and then we deal with it back in our laboratories. Sometimes we take subsamples, in which case we're going to remove the outside, take some plugs, and then freeze it. So that's another step where if our tools are not properly contaminated, decontaminated or sterile, then we can introduce contamination. So what are the potential sources of contamination when we're thinking like of core samples? Well, first, the drilling fluids and seawater, because the core has to travel through seawater. And drilling fluids are made with sepiolite and seawater. So that's source number one. Then it comes on deck and we have to manipulate it. So any type of sample processing. So if our hands or our gloves are not sterile, if the air is not sterile, then we can introduce contamination. And things that we would assume are clean and free of contamination, like the DNA extraction kits, the PCR enzymes, all the material that the reagents that we are using are not free of contamination. So every extra step we add to our protocols is a potential source of contamination. When we are on board, especially the drilling vessels, there are some ways we can test for contamination using fluorescent microspheres or tracers, and these are pumped directly in the fluids. So any part of the sample that will be in contact with the drilling fluids should be positive for these tracers. So when we test for contamination on board the ship, usually we're going to take 3 samples. The first sample will be the outside of the core, and this one should be positive. So here the little green spheres are these microspheres. But when we process our samples, we know that there's probably contamination on the outside. So we remove the outside of the core and take only the inside. In which case drilling fluids should not have penetrated all the way inside the sample. So this should be our negative. And if this one is positive, it means that our sample is not good to be used and is contaminated by drilling fluids. And we often take also, if we're working with basement samples, we'll take some small rubbles in the core liner or even fluids, and these ones should also be positive. And this is very useful when you're on board and you want to say, okay, this sample, we can't use it for future applications. It has been contaminated by drilling fluids. But it doesn't say anything about post-processing contamination that we introduced when we were playing with our sample to get our data. So I got this it's a USEP grant novel project idea. It was novel project idea where I was like, you know what? IUDP has a lot of samples in freezers that are labeled MBio. well, are these contaminated? And can I help other researchers who want to do microbial analyses, like help them at least figure out what the contamination, what are the sources of contamination, how we can prevent it, and how do we deal with it? Because it's not because the samples are contaminated that we need to throw them in the trash necessarily. And it has been published in this year in 2025 in Access Microbiology. So we had 3 questions. The first one was, what are the most common contaminants in drilling samples? Are preserved IODP samples collected on previous IODP expeditions that are labeled MBio, so for microbiological use, are they good to be used for microbiological studies? And the third question was, how much contamination can be found in IODP sediment and basement samples? So the methodology that I use. So the first thing I did was I did a literature review where I was looking for all the papers that cited IODP samples. And in my case, I just focused on 16S ribosomal RNA gene amplicon sequencing, because if we start using metagenomics, it's very hard to know what is contamination. But if we use a marker gene that is found in all bacteria, and that's usually what we use to determine who is in the samples, I figured that's a good place to start. So in my literature review, I had about 66, I think, papers that I found, but I could only keep the ones that fulfilled my 4 criteria. There had to be any kind of controls that were used throughout their process to assess their level of contamination. So oftentimes when we sample, we're going to get seawater, drilling fluids. We're going to do extraction blanks, in which case we do a DNA extraction without any sample. We're going to do PCR blanks, where we do a PCR and amplify our gene without adding any DNA, or just the water that was used for the PCR. So these studies, they had to have at least one control. The sequencing, the 16S genes, ribal RNA sequences had to be generated using Illumina technology, just because it has a lower error rate than some other technologies. The sequences had to be from natural samples and not from enrichments, because I wanted to see it to reflect the pristine or original sample. When we do enrichment, we're purposefully trying to get something that will be the end product will be biased towards what we're trying to enrich. And the last one, the raw reads had to be available in the NCBI SRA database. There were a lot that were available in MGRAST, but their pipeline to download samples was just too tedious. It was one sample at a time, so I decided not to go for that. Unfortunately, a lot of the early census of deep life like studies could not be used, and those were like a lot of from sediment, because they used Roche 454 sequencing technology, which has a lot of errors. And the bioinformatics softwares that I was using were looking for amplicon sequence variants, so just single sequences with 1 or 2 differences, rather than clustering the sequences at, for example, 97% similarity, like we do with the operational taxonomy units or OTUs. At the end, out of the 60 studies that I identified, only 4 passed all 4 criterias, but they encompassed a total of 313 samples. So I had my data, downloaded them from the SRA database. I already told you I ignored MGRAS. It was just too complicated. And then I used Data2 to analyze these data. They have a big data pipeline, in which case you can have a whole lot of samples analyzed at the same time. But I already showed you these were from different studies, and they all had they were all looking at the same region of the 60ness, but but they did not have the same length of reads, same quality of reads. So I had to analyze these sequences in little batches. So I had to divide them into groups where in some cases the entire sequences that were available in the databases had high quality. So I didn't need to do any trimming to remove the low quality sequences. I had one group of sequences where the forward primer was high quality, but the reverse was was pretty much low quality. So I had to cut to very short, 150 base pair. And then the last group, they had very high forward quality, but and pretty high reverse as well. So but in the end, I was able to still combine all of my amplicon sequence variants. And and now I had these groups, but these were all now together, and I resplitted the samples based on the type of control they were. I had 313 samples, so I had to, instead of separating them by papers, I separated them by what kind of control was it. So we had some that were scientific ocean drilling associated, so drill, oil, lubricants, like things that were sampled on the ship that are involved in ocean drilling. Some that were seawater and drilling fluids. I put them together because drilling fluids are a mix of seawater and cypulite. And then the laboratory contaminants, where someone had like the air, that lab air, molecular reagents, extraction, all the PCR reagents. So I have like, and I will use the same color coding for the entire presentation. So we have the drilling controls, DNA are in purple, DNA extraction, PCR blanks are in green, lab air in orange, light blue are the drilling fluids, and dark blue are seawater. And at the end, out of the 313 samples, I had 13,960 amplicon sequence variants. And out of these, there were 29 samples actually that gave 0 sequences. So that means that these negative controls, and you can see they're all here in the PCR blanks, they actually did not amplify anything. They were very clean controls. So what we see here is that the number of reads was quite varying, but so from up to like 30,000 down to 0. But if we look at the number of amplicon, so that was the total number of reads in the study. And here it's for each the same order of the studies. We can see that that's the number of amplicon sequence variants. So the number of sequences that were unique or at 2 base pair difference for each of them. And what we see is that in general, the number of sequences that were sequenced, the reads, did not necessarily impact the number of ASVs. So we can see here, even if we didn't have a lot of reads, maybe 10,000, it was still about the same number of ASVs than it was even if you would have sequenced 30,000. So, and that's true for almost all of the samples. So now I had my ASVs, and I assigned taxonomy to identify the source of contamination, with which I built a database of contaminants, and then I tested the database on different samples. And I will show you the results of that. So what are the most common contaminants? And here it's the number of reads, well, the relative abundance of reads for all of the controls that are drilling contaminants and negative controls. So the negative controls, the PCR blanks are here in green, and in purple, the drilling controls, and orange, lab air. And there's a little bit more variability in the DNA extractions and PCR blanks. But one thing that you can probably all notice right now is that a vast majority of these sequences are Burkholdereales. This like forest, like it's like lime green here. This is the relative abundance of these reads. Now, if we convert this into amplicon sequence variants, well, we still see our Burkholdereales, but it's a very, very small part. So it looks like even if there were just a few sequences of Burkholdereales, either they were present in higher abundance, or there could be some bias with the amplification that once they start being amplified, maybe the primers love their sequence more or something like that, there is an over-amplification effect. But clearly, Burkholdereales, if I would find Burkholdereales in my sediment sample, and this is a very, like, it's one of those bacteria that is found everywhere. But if I were to find it in my deep sea sediment, I would probably start questioning if it is true to the samples or because it came through various other sources of contamination. Now, if we look at the seawater and drilling fluids, we see that it's much more uniform, but pretty much all the water samples look very similar to each other. And here in this case, if we look at the ASVs, it looks very similar to the relative abundance of reeds, as opposed to here where the relative abundance of reeds was very different overall to the ASV sequences. So, but there's also way more microbes in seawater than there are in drilling oils in the air. So the lower the biomass, the higher the likelihood of over-amplifying contaminants become, which can be really a big problem when we are talking about samples that have 100 cells per centimeter cube. I know presenting tables is not always the most fun things when we're looking at presentations, but here I'm just putting the occurrence of the most abundant ASVs, and the most abundant ones like 52, 54 out of 313. So it's about in 1 out of 25 samples, we would find these Burkholdereales. And I showed you Burkholdereales was the light lime green. It's also dominating this list of the 25 most abundant contaminants. And if we look in terms of number of sequences it was found in, we can see that 2 million times this sequence was found across all the samples. And just among the top 25 here, we can see that the number 25 is found like about 20 times fewer than the top one. And again, we can see that my favorite, Burkholdereales, is also dominating this table. So what are the most common contaminants? It depends what the contaminants you're looking for, but if they are the laboratory contaminants, I put them here in purple. If they are from seawater, I put them in blue. And if it's in gray, it's because they were found in both. And that just highlights the overall ubiquity of some microbial groups. Here, I'm using the order level, and you may be questioning why I'm using order level. If we're thinking in terms of phylum, it would be like proteobacteria. Class would be gamma proteobacteria for a lot of them. And then at the order level, we can start getting a little bit more specific. But if we go too far, the length of the 16 SPCR products that we are using does not necessarily allow us to go very far in the taxonomy. So I decided to keep the order level. But I did try it with other ones, and this one seemed to always capture the contaminants in my other tests that I did. So now we have our answer to what are the most common contaminants. So now we have these, what is the fraction of contaminants that was found in all these controls versus the seawater? So at the top here, we have the controls that were the drilling fluids and the PCR blank. And if in green, we have those contaminants that were found both in seawater and the contamination controls. So you can see in the blanks and when there shouldn't be any microbes, basically, it's very much dominated by these contaminants, as well as the pink ones are the common contaminants, and in black would be other. So you can see that if we look at these controls, that just by using these 30 groups of microbes, we can capture almost every contaminant. In the seawater, we can also, it's not capturing as much, maybe 70%. All the gray one is the variability between the seawater samples. But blue and green again, so dominant in seawater and found in both was making up about 70%. So again, if we're dealing with seawater infiltration, it's quite possible that, especially if we're looking at samples that are in the like not more shallow of the sediment, it's quite possible that we could find these microbes and they would belong there. So what I was suggesting in the paper is, as long as we label our like sequences as potential contaminants or potentially from seawater, I think it is correct. As long as we handle and that we acknowledge the fact that it's quite possible that low biomass samples are contaminated. So I addressed what are the most common contaminants. Now, I mentioned to you there are some samples in freezers at Kochi, in Bremen, and in College Station that are in the freezers labeled MBio, and they were collected on various expeditions for microbiologists to use. Well, are these actually suitable for microbiological studies? So here I have just the map of the IODP, the Ocean Discovery Program, where we have like all the expeditions that occurred. And these are the ones that had at least one objective that was microbiology related. So for example, Brothers Ark Floss, one of the main expedition objective was to assess the diversity, extent, and metabolic pathways of microbial life in an extreme metal-oxic and acidic volcanic environment. So this one, there are samples that are available, and they were taken most likely by microbiologists because there was a microbiology objective. But there are other here expeditions where right now there's no yellow dot, but we do have samples that are labeled MBio. And we don't know if they were collected by a microbiologist or a geologist or an engineer in some cases. So can we use them as microbiologists? And here's a list, and I need to add there's a few more expeditions after this, but of the expeditions that are labeled on these repositories websites that say we have MBio samples. And here we have, like, if there was a microbiology objective, I put a little X. So we could go in those Nantroces expedition and get MBio samples, but these were not for microbiologists. Is it clean? Is it not clean? I'm not sure. And that's what I wanted to test in this. So I selected samples from 3 expeditions, and they were all related to they were all in the Japan Trench, and I was taking these samples for 1, do the contamination testing, and 2, we were also trying to these have been frozen at -80. We were trying to see how much of these microbes can actually survive the freezing, and we did some other incubations afterward trying to revive these microbes. But I ended up with a range of samples. Some were plugs, some were whole round, and they also varied in the depth. Because as if you remember earlier, I told you that the deeper samples often have fewer cells than the samples that are at shallower depths. So would this one be more easily contaminated than this one, for example? So what did we do? When we got a whole round, we worked in our anaerobic chamber because these were like in anoxic conditions. So that's the little gray box here. We got the whole round. We removed the outside, which we know is most likely contaminated because it touched the core liner, the drilling fluids. And then we did a DNA extraction, amplified the V4, V5 region of the ribosomal RNA, 16S ribosomal RNA gene, and then we sent it for sequencing. And we got this. So here we have our 6 samples, and I'm not even telling you what's in there. I just classified all my sequences based on my little database that I showed you with my 2 tables, whether they were in like seawater influence or drilling contaminants, likely laboratory contaminants. And here I put identified with blanks. When I did this, I also generated my own blank, and but all the blanks that I identified in my blanks were in my database that I generated, except that one sequence. So pretty much the database, even though it's quite rudimentary, it only has 30 orders of microbes, it can capture all the contamination that was present in my all my PCR and DNA extraction negatives. So I think it's still quite robust, even though it's not very extensive. And if we look at the amount of contamination, so here we want green, this lime green. It's there were 2 samples that had none to very little contamination, but 3 of the samples had up to 95% contamination. All the contamination it wasn't from the seawater, it was mainly from the laboratory because these were all low biomass samples. And the ones that had no contamination were whole round. So if you remember at the beginning, I put little numbers on the arrows. So the less amount of time the sample is touched, the better it is. And one of these was actually a wash core. And it means they took the whole round, they cleaned it up, and then they froze it. And we got it. And then I wasn't sure what had been done to it. So I cleaned it up again. So it had been removed, like contamination has been removed pretty much twice. And we only kept the very little middle, but it was also very not contaminated at all. And the most contaminated ones were the plugs. So when you start taking your tool and add another step for the collection before if your syringes that you're using to make holes are not clean, and the longer it stands on the bench, the worse the contamination can be. And also this one whole round, which was most likely clean, but from the very deep sample, 850 meters below seafloor, that one was also quite contaminated, which is probably because it had the lower biomass. Therefore, if you just have one or few cells that are contaminating, they can overtake everything else. So what it means, contamination is more likely to occur when the sample is more heavily manipulated. So if you have a plug, you just added one more step where you can introduce contamination. When the coring strategy is disturbing. Oh, I didn't talk about coring strategy. So the ones, oh yeah, this one, that was very deep. They used rotary core barrel, rotary core barrel uses more drilling fluids, and it's more disruptive than if you use APC or XPC. So depending on the drilling technology used, then the integrity of your sample may be damaged. And if the biomass is lower, then it can also impact the level of contamination. So now I tested some IODP samples, and really the answer, are they usable for microbiological studies? It depends. It depends on what the samples were, how they were like collected, how they were manipulated. So I would not be comfortable going back to this long list of samples and say yes, yes, no, no, no. I think it's a case by case testing that has to be done. And now I wanted to see through other studies how much contamination can be found in other IODP sediment and basement samples. Because when we're thinking, how do researchers handle contamination? Well, usually they'll use their own PCR and DNA extraction control, and some of them use a statistic. Okay, if it's found X% in my sample, but like a much lower percentage in the control, it's not like I can still keep it. Or they set like statistical thresholds. Some, like me, are more stringent, but they will take only things that were found in their personal control and then remove it at the ASV or level or other level. So mine is quite stringent, but I wanted to see, like, there are studies that have been published and that have no control. So, because it happens, life happens. Sometimes there can be sequencing issues. I showed you 29 of the negative control did not provide any sequence reads. But does that mean that all your samples are pristine and uncontaminated? Or does that mean, well, you had a really good negative control that day? And sometimes also samples can be lost. So I wanted to try to make something easy that could be used for people that are maybe not experts in 16S ribosome RNA sequencing and analysis, and see, so they could at least acknowledge, okay, I got my sequences, but this part may be contamination. This part is probably good. So I found two studies that were published and unfortunately did not use controls. So the first one was a study where they did the Chicxulub. They went to where the crater that failed that killed the dinosaurs, and they used a platform like this for their drilling, and they drilled at a single site, and they used rotary core barrel. So when I see this, I'm like, well, I'm pretty sure that they're like, so this platform does not have an MBio lab, most likely. That was like it was on the JR, or even better, the CHIQ. And I know that rotary core barrel is more disruptive. So I took their data and I labeled them. If the sequences were found in seawater or drilling contaminants, then it's blue. If it's laboratory contaminants, like this famous Burkholderia, pink. And if I wrote likely endemic, if it was not found in my database, so it's probably that it was present in their sample. And we can see here that there was between 15 and 97% of sequences that were potential contamination. And because like it's quite possible that some of these seawater sequences are found in the sediment. But there were a lot of them that were also maybe from the like all those laboratory contaminants that I've discussed about, whether it's DNA extraction, manipulation, PCR. But there was no real trend between like the depth. So here at 500 meters or 1200-meter depth, there's about the same amount of potential contamination throughout. And then I found another study which was in the South China Sea, and there they did multiple sites, but I just used the depth of the sample, and I did the same thing. And they use different technologies, and it was depending on the hole, and usually there were more contaminants in the holes that was rotary core barrel versus the other APC or XPC. We also see in here that the majority of the samples have quite a large chunk of sequences that are most likely endemic, and there's much fewer potential contamination. And actually, like 52 out of 60 samples had less than 50% contamination, which I think is pretty good. Especially if you know you can spot your contamination, you can still use all of their other data. And all the samples had more than 30% of all the sequences that were likely real sequences. So I'm going to summarize what I've just told you. So with the example of the JR, we identified the potential sources of microbial contamination, whether it's drilling fluids, seawater, sample processing, or even when we're back in the lab, DNA extraction, PCR amplification, and sequencing. I built a database of potential contaminants that are found in scientific ocean drilling data sets. And we made this so now if you don't have a control, you can use it. And I'm talking here about scientific ocean drilling, but any sort of drilling would actually, or samples with biomass would be most likely contaminated with the same at least from the control, not the seawater, but all these other ones would be most likely the same because we breathe the same air, our labs are using the same reagents, etc. I also looked at the contamination level in preserved sediment microbiological samples, and we could see that the least manipulation, the better your chances are of not introducing contamination. But also the drilling technology can make a big difference. And then we identified the potential. We looked at other published studies that unfortunately did not have negative controls to see what was the proportion of sequences that were potential contaminants. And really what we have to look for is the drilling type. And now that the JR is retired, there's going to be a lot more of those mission-specific platforms. And so I think it's going to be important to look at the type of drilling that is going to be used, and if there are other options that can be used so we can preserve better microbiological samples, sample manipulation. There was some sort of relationship with depth, but that was mainly due to the type of drilling that was used. As we go deeper, sediment is more compacted, then you have to use more disruptive drilling technologies. And the biomass, if you amplify sequences from a sample that should be technically sterile, like your DNA extraction blanks, there's not a lot of biomass, but because of that, when we amplify, we're going to over amplify this contamination. And I would like to thank InterRidge for inviting me, but also this work was done with 3 of my students. Milena, who I saw is connected here today, and Kate and Jordan, who graduated last year, were also part of this. And with that, I would like to take any questions.
Thank you very much. We appreciate having you. Yes, feel free to type your questions in the comment box, or please also raise your hand or unmute yourself as well. I'll kick us off if that's okay. What was the biggest surprise that you had in this meta-analysis?
I think one thing that I would like to test further is why Burkholderia is so much over amplified. I don't know if it's over amplification or it's really that it's in the samples. And I think we could do some tests where we actually like put a certain amount of Burkholderia and like have more of a control community, because I was very surprised at how dominant it was in those samples that were not supposed to have any microbes in them.
What about Ralstonia? That has been
Mhm.
a source of discussion, in past CODL studies and also in the Sheik et al. Paper
Mhm.
as well. What are your thoughts, or would you
That
just?
was the one that was the, if we go
Yeah.
back to the my table, Ralstonia is at the top. It's at the top like 3 times. Yeah.
Do you think that's from the kit, or do you think that is from?
Yeah, I don't know. I would have to go back. But yeah, see it found in 54 out of 52. And here, like, yeah, 2 million sequences. And so I think, yeah, we would have to check exactly where it's from. Um, but I think it's everywhere. That's the thing. I wouldn't be surprised that we breed it every day. It could be in the kit. I know it was one of the bigger ones in those papers where they only tested DNA extraction kits.
Should we just throw out Ralstonia altogether?
Yeah, burn it to the ground. Uh,
Meaning, if we see it, just remove it. It's
I think so, or label
in
it like
olden days, we used to do that with cyanobacteria, but we learned that there was a purpose to the.
Mhm. One way we can do it is to do like some sort of a phylogenetic tree where you would have like Ralstonia from the air, the DNA extraction kit from sediment, and see if they all group together or if they are just if it's the same exact sequence, then we don't know, but most likely not from sediment.
The Steve Goldstein has a great question as well. Do people
Yeah.
use clean labs either on ship or land to minimize contamination? And if so, what type?
Yeah, so if we think like on the JR, there is a biosafety cabinet, which is clean, but they put it in the 4-degree room, so it makes it hard to work there for long hours. They also got a bench top where that is pushing the air up, limiting contamination. So there are, uh, but that's why, like, if we can just put it in a bag, freeze it, and deal with it in our individual labs, it's better because we all like have labs that will have like negative pressure and all the proper equipment to deal with to limit contamination, but it's still there. And if you order your kit, the extraction kit, you can open it in a clean environment, but if it's already contaminated at the where it's made, then we have to deal with contamination. I'll put this one there.
Uh, Melody Lindsay asks, what are your opinions on the lists of common contaminants of PCR DNA extraction kits and their potential use as a catch and remove all during bioinformatics screening out of contamination? And could we use this amazing work of yours as a database of sorts to bioinformatically remove contaminants from deep biosphere sequencing data?
Yeah, so it's it's about the same way that I did mine. Mine was just more specific because you also have the seawater. But if you could, like, that's, I just wanted to try to make it simple. But if you have your list of ASV, and you can easily just label those as potential contaminants and call it a day, in your bio when you finish your bioinformatic pipeline. And if you're working with sediment samples, then you can throw in the seawater as well. It's more tricky because there could be some of these in sediment because of water infiltration and everything. But I was just trying to make it easy for people. Now you have a list. If you want to use it to just label potential contaminants, you can still keep them, or like in your supplementary figures, like include everything. But I think it's important to at least acknowledge it. It's a fact that we have to deal with contamination. Unfortunately, it is uh, the the ships are, yes, they're equipped, but I would say the Chicky was probably a little better than the JR was, because there were lots of microbiologists involved in the designing of the ship. But it is an issue we just have to acknowledge and deal with it. But yeah, if the list is in the paper, you can just use this and be like, okay, they're probably contaminants. But I still highly recommend you, Melody, that you keep doing your negative controls. And to everybody, it doesn't mean you should not talk not do your blanks. Now that we have a list, you should still do them.
Yeah. Can you speak to if you have any experience in this that maybe someone on the talk, maybe on the audience may find it helpful, the coach system or the coach system? Um,
Yeah, actually, I think I had a picture last week. I don't remember where it went, but yeah, it's a box
mhm.
that pushes air, and I think also it has some sort of like static or electric, like to charge the air differently. I'm not sure.
Yeah.
But when we used it, we actually measured the number of particles that were in the coach box versus the air outside, and there were like tens of thousands of particles in a minute on the outside of it and less than 50 inside. There's still 50 particles if you're, it's not foolproof, but it's still better.
So if we have an extra $20,000, it'd be useful.
Yeah.
Maybe we should be writing that into our budgets. Well, thank you so much. Again, if there are any other questions, sure, feel free to reach out to Jessica as well. And once again, if you have any suggestions for future speakers, please let us know as well. But we look
And
forward to
maybe next
seeing.
time I'll talk about viruses, but
Yeah.
yeah.
Welcome back anytime. Uh, Next webinar is November 5th with Helene Lemavelle. Thank you all.